biosafety and on matters microbiology. She is an expert in these particular areas, and she will be able to share with us much about herself. And in a short while, she will take it away and start the presentation. Welcome, Madam Michelle, and I hope you are able to hear us from this other side. Are you able to hear
Hello.
us, Madam Michelle?
Yes.
Thank you so
Yes,
much.
I'm able to hear you.
Yeah, we wish that you put your presentation on the slideshow mode so that you can maybe greet the participants, and then you can do further introduction of yourself better than the way I've done it, and then so that our members can get to know you well, so that you can as well proceed and start the presentation. Thank you. Welcome.
Thank you so much, Arthur, for the introduction. Yeah, I won't add much to that. I'm a medical laboratory officer. I have worked with Metropolis for ongoing 5 years now, and I am primarily based in the microbiology department, and I'm also the organization-wide safety officer. So I'm also taking up on the safety role. Now, yes, my background is in Bachelor of Science, Medical Laboratory Science, and that is what I've practiced since my graduation. So yes, the purpose of the presentation today is just to explore the bacteria identification methods. So we will see what methods are available here, from the traditional ones to the more automated methods and even the novel technologies that we have. So I don't know if this is my cue to start. Arthur, please confirm.
Yes, go ahead.
Okay. So yeah, that is my introduction. So our objective today will be to understand some of the basic ways bacteria are classified. We'll then transition that to the phenotypic identification techniques, where we will differentiate between manual, semi-automated, and automated phenotypic identification systems. We will also highlight some advanced identification tools, and then we will summarize the key considerations in selecting appropriate identification methods for clinical microbiology labs. Then, after this presentation, I hope that I shall have laid groundwork in the understanding of antimicrobial susceptibility testing. So from this presentation, we'll be able to seamlessly do another PPT on antimicrobial susceptibility testing, which precedes identification. So bacterial classification. We can classify bacteria in different ways. Some ways of classification are relevant to your laboratory settings. Some are relevant in research settings. So these are just some of the ways we can classify bacteria. So we have in terms of morphology and arrangement, we have in terms of their staining properties, bacterial growth and propagation requirements, their cultural characteristics, biochemical reactions, their antigenic structure, and base composition of bacterial DNA. So just like I've said, some of these classification are more relevant to clinical settings, others are relevant in industrial microbiology, and so on and so forth. So I'll just highlight on the main ways that they are classified and what is really relevant in clinical microbiology. Yeah, so some like base composition of bacterial DNA will be used in metagenomics and
Uh, Madam Michelle, excuse.
yes.
Are you able to move the slides, because we are still on the first slide? Are you able to move the slides on our side? We are able to see the first slide only.
Maybe my slide movement is not consistent with what is projected in the screen. I'm able to move my slide. I'm currently on a slide. The title is Bacteria Classification. So I don't know if you're in the same slide.
No, we are on the first slide, Lab Medicine Society of Kenya.
So what I'll do, I'll stop sharing my screen and then share again to see if we can move more consistently. Is that okay?
Yes, that is okay.
So can you see I'm on slide for bacteria classification?
Yes. Thank you.
Yeah, that we have discussed. So let's discuss bacterial classification in terms of their morphology and arrangement. So when bacteria are visualized under a microscope, the following morphology are seen. So you can have cocci, you can have bacilli, you can have coccobacilli, you can have spiral bacteria, and even curved bacteria. So yeah, that is classification in terms of morphology and arrangement. So this is just a picture. I hope you can see the picture. Some of the shapes, the morphological shapes of bacteria: Coccobacilli, bacilli, diplococci, tetrads, streptococci, staphylococci, and also the comma and the spiral shaped. Then we can also classify the bacteria in terms of their staining properties. So we can have where we do the major staining property that we'll check is the gram stain staining property for this particular PPT. So the gram staining reaction is used to identify pathogens in specimen and cultures by their gram reaction, where you can have either gram positive or gram negative. And this you combine with the morphology that I have just shared on the previous slide. So the gram stain reaction in combination with the morphology, then you have you can classify them in terms of gram positive or gram negative bacteria. So for gram positive bacteria, they stain dark purple with crystal violet and are not decolorized by acetone. So some of these bacteria are Staphylococcus, Lactobacilli, Streptococcus, Clostridium, and even Corynebacterium. For gram-negative bacteria, they stain red because after being stained with crystal violet, they are decolorized by acetone and take up the counter stain, which is neutral red or safrarin, depending on your clinical setting. And in this category, we have Neisseria, Klebsiella, Haemophilus, and Brucella, Salmonella, among others. So in terms of bacterial growth and propagation, yes, you can have the temperature requirements, the atmosphere requirements, or the oxygen requirements, and even the salt tolerance. That is in terms of their need for growth and propagation. So in terms of temperature, you have your psychrophiles, your mesophiles, and your thermophiles. And those are just the temperature ranges that these specific bacteria can tolerate. And then you have your atmosphere requirements. So in addition to varying requirements for optimal growth temperature, bacteria also differ in their use of oxygen for respiration. So you can have aerobic bacteria that choose oxygen as a terminal electron acceptor during respiration. You have microaerophiles. They also require oxygen, but at a lower concentration. And then you have anaerobic bacteria. So they can they use inorganic electron acceptors that also require the use of oxygen, but at lower levels than naturally occurring in the environment. So in terms of the growth requirements, you could also have the soil concentration requirements where you have your halophiles, your osmotolerant organisms, and even your non-halophiles. So I didn't include that, but you can just note that as well. Just give me 2 seconds. Let me adjust something. Yeah, so basically, that is how we are classifying them. And then I now want to transition into how we identify them. Okay, so that will lead me to the next slide. Just a minute, bear with me. Yes. So I hope you can see my next slide, which is the method used to identify bacteria. So the accurate identification of microbes is essential for scientists involved in many areas of applied research and industry, which ranges from clinical microbiology to food production. So there are many criteria for the division of the abundance of methods used in the area of identification of microorganisms. However, for the purpose of this presentation, I would like to categorize to use two categories or two techniques. So we have the indirect techniques or methods, or the direct techniques or methods. So for the indirect techniques, these are culture dependent methods. Okay, so these are methods that will require you first culture and isolate an organism before you proceed to use them for your identification purposes. So the isolation and culture of microorganisms and the determination of their various phenotypic characteristics. So where you can have different indirect techniques like the chromogenic media, serotyping techniques, biochemical techniques, and even protein analysis, where we are going to mention mass spectrometry. So remember, indirect techniques will require you to conventionally grow the bacteria before you proceed to use them for your identification. For direct techniques, these are also culture-independent methods. They may be used to identify specific microbes in a mixed population, as well as identify non-culturable microbes. So you have microscopy, and here I will also include molecular techniques. So microscopy techniques will be by visualization of characteristic structures and for organisms in the viable but not culturable state. So this is where you'll now use the classification slide, where we were classifying them in terms of their morphology and arrangement. You can make your observation from such visualization from a microscope. And then you have your molecular techniques, which will delve into the genetic makeup of the bacteria. So the direct techniques will not require you to culture. They will help you identify, but you don't need to culture. So like microscopy techniques, when you're talking about the viable but not culturable state, you can bear witness that in HVS has lactobacilli. So these are organisms that may have a very strict culture requirements, and therefore you will just need to maybe use the microscopy techniques to narrow them down depending on their morphological presentation on your microscope slide. Yes, so that is what we will do. So let me mention the chromogenic media, then I'll mention the serotyping, and then we'll look at the biochemical techniques, and then we'll look at mass spectrometry, and then from then we'll look at the molecular techniques. So the microscopy techniques, I will not really delve much into it. It's basically you stain and you check in your microscope. So yeah. So for the first indirect method that I've listed, the chromogenic media. So these are basically they're a type of culture media used in microbiology that incorporates chromogenic substrates. So these substrates react with specific enzymes produced by microorganisms, resulting in a distinct color change that allows for easy identification and differentiation of bacterial colonies. So this simplifies and speeds up the process of microbial identification, often eliminating the need for further biochemical tests. So the principle of chromogenic media is that a chromogenic substrate is split by an enzyme that is characteristic of the target organism into a sugar component and a chromogen. So in the presence of oxygen, the chromogen forms a dimer that colors the broth or typical colony. Okay, so that is just the basic principle. So from the chromo, you'll relate it to color change. So you'll have specific media from different manufacturers that will give you the specifications on how to use their chromogenic media, the color change you expect if you are narrowing down to a specific organism. They'll even have the charts for those color change and how you can basically narrow it down and ID an organism from it. So you could even have a chromogenic media specific for E. coli 0157. So depending on the colony the color of the colonies, you're able to decipher if it is indeed 0157 or if it's another it's another strain of E. coli that is not 0157. So they are very specific for that organism, for the target organism. Okay, so the chromogenic substrate is very specific for the target organism that will produce a color change that will help you negate the presence of that organisms, or you'll also check for if that organism is absent. So you could have that, like I've said, you have maybe E. coli 0157, or you want to check for methicillin resistant Staphylococcus aureus. Yeah, so this you'll utilize this media for that purpose. So it negates further biochemical tests. So if you are narrowing it down to a specific organism, the chromogenic substrate is specific for that organism. So then you don't need to do other biochemical tests that will now help you again narrow down to an organism because it's already specific for that organism. Then you have serotyping. So serotyping is a method used to classify bacteria based on their surface antigens, which are molecules that trigger an immune response. It is a valuable tool for identifying different strains of bacteria, understanding their pathogenicity, and tracking outbreaks of infectious diseases. So yeah, you have your antigens. Bacteria possesses surface structures like lipopolysaccharides, capsular polysaccharides, proteins that can act as antigens. And then you have the antibodies or your antiserals. So the serotyping utilizes specific antibodies that bind to these bacterial antigens. When antibodies bind to their corresponding antigens, a reaction like agglutination or precipitation occurs, allowing the lab staff to identify the bacterial serotype. Then you have, it is very important, especially in our clinical microbiology settings, in the differentiation of strain types, okay? So serotyping can differentiate between strains of the same bacterial species, including those with varying virulence or public health importance. I've just mentioned E. coli 0157. So there are many strains of E. coli, some that are pathogenic and some that are non-pathogenic, some are more virulent than others, and some you'll consider of public health importance. Now, most of the identification methods will not commit to the identification of these organisms to the strain level, some even to the species level. They will tell you to complement your identification with serotyping methods. So this is it's a very powerful tool when you're really keen on trying to know the bacteria to the species and even to the strain level. Now we are moving on to biochemical techniques. So in microbiology, traditional identification methods rely mainly on cultivation proceedings, employing various media to enumerate, isolate, and identify specific microorganisms. So phenotypic identification methods usually incorporate reactions to different chemicals. So in this category, we're going to know the manual phenotypic identification methods that are available, the semi-automated and even the automated phenotypic biochemical identification methods. Just a minute. Yeah, so what do we know about phenotypes? Because somebody might be asking, you might be wondering why phenotypic, why are these called phenotypic identification methods? So the phenotype of a microbe can be viewed as an observable characteristic of that organisms. So that could be their morphology, the consumption or production of certain metabolites, or the capability of growth in an environmental niche. So these particular methods will utilize the phenotypic differences of different bacteria to help you narrow it down to a specific bacterial ID. So you have your manual phenotypic identification methods. So in this, you have catalase, coagulase, bacitracin, optochin, bile, esquilin, TSI. These are some of the mostly used manual phenotypic identification methods. We will also discuss the semi-automated phenotypic identification methods and the fully automated phenotypic identification methods. So like I've said, you're going to see their morphology, the phenotypes you're going to see their consumption or production of certain metabolites that now help us say that this particular organism is this genus and species. So the manual phenotypic identification methods from gram-positive bacteria, you have catalase tests to differentiate between staphylococci from streptococci. You have coagulase tests that are used to identify Staphylococcus aureus. So it differentiates Staphylococcus aureus, which is coagulase positive, from coagulase negative staphylococci, such as Staphylococcus aureus and Staphylococcus saprophyticus. You have novobiocin to differentiate Staphylococcus saprophyticus from other coagulase negative staphylococci, where S. saprophyticus is novobiocin resistant. Then you have bacitracin test to differentiate Streptococcus pyogenes, which is bacitracin test positive, from other beta hemolytic streptococci, which are resistant. Then you have optochin test to differentiate Streptococci pneumoniae from other viridans streptococci. Then you have your CAMP test to differentiate between certain organisms such as Streptococcus agalactiae. So CAMP test, I would say, is the gold standard for identifying Streptococcus agalactiae. So I will not delve into the principle of it and the final outcome of a CAMP test, but I'm just listing it here. Then you have your DNase test. So this test is used to differentiate Staphylococcus aureus, which produces the enzyme DNase, from other staphylococci which do not produce DNase. It is particularly useful if plasma is not available to perform coagulase test, or when the results of a coagulase test are difficult to interpret. So I've just listed some of them, not all. So yeah, just for the purpose of the presentation, those just are some of the few phenotypic identification methods for gram-positive bacteria. Then you have the manual phenotypic identification methods for gram-negative bacteria, and I've specified the big family in this category, the Enterobacteriaceae. So here we have citrate utilization test. The test is based on the ability to use citrate as its only source of carbon and ammonia as the only source of nitrogen. So you'll have Klebsiella and being positive and E. coli being negative. You have indole test for indole production, testing for indole production for differentiating different species of Enterobacteriaceae, E. coli, Proteus vulgaris, Morganella boganii, Providentia. Those are mostly known to release indole and are indole positive. And then we have oxidase test. It's not really used to narrow down your Enterobacteriaceae family, but it will help you negate Enterobacteriaceae or help you narrow it down to other bacterial organisms such as Pseudomonas, Neisseria, Vibrio, Pasturella species. So these organisms are able to produce oxidase enzyme. Then you have urease test. Testing for urease enzyme activity is important in differentiating Enterobacteriaceae. So Proteus strains are strong urease producers, and Salmonella and Shigella do not produce urease. Then you have your TSI test. Employing triple sugar ion agar is designed to differentiate among organisms based on the differences in carbohydrate fermentation patterns and hydrogen sulfide production. Carbohydrate formation is indicated by the production of gas and a change in the color of pH indicator from red to yellow. So these are some of the manual phenotypic identification methods that I know some of you are using in your laboratories to help you narrow down your bacterial ID. So these are some biochemical, this is just a biochemical test chart. So once you do your gram stain, you see that it's gram-positive bacteria, then it informs you of what biochemical test to do next. So you will see the, let's take the cocci, where you're using catalase test to differentiate between Staphylococci and Streptococci. So Streptococci being catalase negative and Staphylococci being catalase positive. So if you have your catalase positive, then you do coagulase. So coagulase negative will be under coagulase negative Staphylococci, and coagulase positive will definitely be Staphylococcus aureus. So how do you differentiate between your coagulase negative Staphylococci? You do novel biosyn sensitivity to differentiate between Staphylococcus saprophyticus, which we've said is novel biosyn resistant, and Staphylococcus epidermidis is novel biosyn sensitive. So for your Streptococci, you differentiate them in terms of your Lancefield grouping, where you'll have different biochemical tests to differentiate between your alpha, beta, and non-hemolytic Streptococci. I think we have also discussed the octochain test, the bacitracin test. We have not discussed the growth in salt requirements, but you can see it from the screen. So the biochemical test for gram-negative bacteria, this is just a simple flowchart that can help guide you in your manual setup to know what to do next. So let's take the bacilli, which are the common bacterial isolate, and you'll use your I've just noted there's an issue with this. There's an issue with this chart. Yeah, so when you get your lactose fermenters, your bacilli, they're gram-negative bacteria, your bacilli, are they lactose fermenting? Yes, they are. Okay, are they fast lactose fermenter or are they slow lactose fermenter? You can group them just the way the chart has grouped them. So are they lactose fermenting? You can ask yourself, is my isolate a lactose fermenter? No. So what do I do next? Oxidase test to differentiate between Pseudomonas, which is positive, and other other organisms, including Enterobacteriaceae that could be that are oxidase negative, and then you'll proceed with other biochemical tests to help you narrow them down. Again, this is just this is not to generalize everything. You also have to know what site is my isolate from. What site is my isolate from? Is this bacteria a common pathogen in this site? So those are some of the decision-making checkpoints that you'll need to help you in bacterial identification, especially in your manual setups. Now, let's go to the semi-automated phenotypic identification methods. So I would like to mention here API. So API is basically a plastic piece of 20 cupels that contain pH-based substrates, allowing the identification of more than 100 taxonomies. So the cupels and microtubes are inoculated with a bacteria suspension that rehydrates the wells containing substrates. During incubation, metabolism produces color changes that are either spontaneous or revealed by the addition of reagents. So all positive and negative tests are compiled to obtain a profile number, which is then compared with the profile numbers in a commercial database. So until 1992, this method was considered the gold standard, especially in clinical microbiology. It has a significant advantage, being that it had an extensive database, although it had a disadvantage associated with it being time-consuming. So your API, you can see on your right-hand side, that is just the plastic piece with the 20 cupels. So those are the cupels have your substrates. You can't see them properly, but ONPG, ADH. So those are basically the substrates that have been impregnated in these cupels. So what you introduce your bacterial suspension, you incubate, and then the next day you check for color change. So the first one is the APA20E after incubation. So this one you have positive results on all tests, and below you have negative results or on all tests. So the negative one will be typical of somebody maybe trying to isolate Pseudomonas aeruginosa using the APA20E profile, which is specific for Enterobacteriaceae family. So you'll have such a result as indicated in the plastic cupel just above the one that has turned positive. So what do you do next after you've incubated and now you want to identify your bacteria? So it has a paper such as this. Okay, this will be where you'll be doing your readings. So you'll just come up with positive or negative depending on the API insert and what you know as positive and negative. So that is what you'll denote here. So you'll come up with an API profile number. So like this lab tech has come up with the profile number 670 or 4752, okay, from the addition of the outcome of these cupels. And then this particular profile number, now the lab tech has to check it in a commercial database. Let's say if your API provider is bioMiro, so you'll have logins to a database that can help you check this specific profile number amongst the profile numbers of different microbials on their database, and then you're able to come up with an identification of an organism. So on your right-hand side, just below, you'll see this is just a screenshot of the outcome of somebody checking the profile number into a database and then coming up with an organism. So that profile number is 1,444, 555, 257, and there is a good identification of Escherichia coli. Then you have your automated phenotypic identification methods. So here I'd like to mention Vitek. So Vitek is an automated version of the API. Okay, it was first developed in the 1970s, which eliminated the subjectivity of the reading of test reactions. So remember, these test reactions, you'll have your insert for the API that tells you if it's green, you're going to give it a plus. If it's blue, you're going to give it a negative, and so on and so forth. But then there is subjectivity. Michelle comes and reads the API. I'm seeing this is yellow. Somebody else is saying, no, this is not yellow, or it has slightly changed. So we end up with different profile numbers that could essentially lead us to different organisms. So this was eliminated by Vitek. So the Vitek system is useful for the simultaneous bacterial identification and antimicrobial susceptibility testing profile from isolated patient samples. So Vitek is still utilizes biochemical tests. The only difference, it has a colorimeter, which will now do your automated readings, and then you'll have your database comparison. So the Vitek system utilizes specialized cards containing a multitude of biochemical tests, such as fermentation of sugars, utilization of various substrates, and enzymatic activity. So these tests involve introducing a standardized microbial suspension into the card and monitoring changes in color or fluorescence based on metabolic activity. The system used an advanced colorimetry to detect subtle changes in color, providing high discrimination between different microbial species. Then we have the database comparison. The results from the biochemical test are compared to comprehensive database of non-microbial responses. The Vitek2 system identifies the microorganism by finding the best match between the observed reactions and the database entries. Then we have we're going to mention the automation and speed of the machine. The Vitex2 system automates the entire process of inoculation, incubation, and reading, significantly reducing manual labor and turnaround time. So this automation, coupled with speed of biochemical assays, allows for rapid identification of microorganisms, particularly important in clinical diagnostics where timely results are crucial. So just like the API, it is culture dependent, and it is database dependent. Okay, so different manufacturers will do different research and development studies to come up with profiles of many microorganisms, so that when you generate a profile number, you can check in the database in case of API, or it can automatically be checked in a database in case of Vitek to come up with a specific organism from the genus up to the species level. So just Vitek automates this process, but this is just biochemical tests still. So maybe if you see the card, if you see the card, this card, you can see there are some color variations in the card. You have blue, yellow, those are just some of the substrates that have been impregnated in those wells. So the difference between API and Vitek is, like, let's say for you want to identify an Enterobacteriaceae, say using Vitek, you'll have 64 biochemical tests compared to the 20 biochemical tests for API. So Vitek is even more specific and more specific and even a more sensitive results because it has 64 biochemical tests. So if you have organisms that are closely knit or share biochemical reaction, similar biochemical reaction outcomes, with the PI20E, you may be missing some organisms, but with 64 dehydrated substrate, you now have better differentiation of different microorganisms. So these are just some of the other technologies we have here. So it's basically they all use colorimetry. You have BD Phoenix, then you have microscam, Beckham, Coulter, just to mention a few. Then we are going to also mention mass spectrometry, also very culture dependent, so it's still an indirect method. So mass spectrometry, let's talk about it. Research in microorganism identification has evolved mainly by following the strategy of reducing the time required for identification of a particular microbial in routine diagnostics. For a method to be considered successful nowadays, the attainment of results should take a maximum of 24 hours, and then in an emergency, there is an urgent need for this time frame to be even smaller. So the method based on mass spectrometry have gained popularity as a microbial typing tool due to their speed, reduced cost, simplicity, and applicability for a wide range of microorganisms such as bacteria, archaea, and fungi. With advancements observed in mass spectrometry methods, along with new data analysis and processing and visualization tools, our understanding of biological system has also increased, since we could analyze diverse sets of biomolecules such as proteins, lipids, carbohydrates, and amino acids. So several ionization and separation techniques can be coupled with mass spectrometry, such as gas chromatography, matrix-assisted laser desorption ionization time of flight, electromigration techniques, and electrospray ionization. So mass spectrometry, you can differentiate proteins, lipids, carbohydrates, and amino acids. Now, for the purpose of clinical microbiology, we use matrix-assisted laser desorption ionization time of flight, together with the mass spectrometry to help us identify bacteria. So you'll find that for the bacterial ones, for the bacterial identification, this will involve the separation of proteins. Now you'll have for lipids, carbohydrates, and amino acids, you could have technologies now for that could replace your immunoassays and so on and so forth. But for clinical microbiology, we're going to look at MALDI-TOF. So MALDI-TOF MS is the latest next generation tool being used for the rapid identification and classification of microorganisms. It is based on the ionization of the microbial cells with short laser pulses, and then accelerating the particles in a vacuum system using an electric field. After the ionization, a molecular fingerprint in the form of a spectra profile is obtained, which is specific for each microorganism. So this spectrum is then compared to an existing database, resulting in its identification by an automated program. So preparation of sample involves crystallization with a large molar excess of matrix on target plates. So that's basically how MALDI-TOF MS works. It's a patent, and then you'll find now different manufacturers utilizing these patents to come up with different equipment to help us identify bacteria. So on your right, you'll see the Vitek MS equipment. So Vitek MS that uses MALDI-TOF MS. So let's talk about Vitek MS. So each Vitek MS has disposable target slide that has unique barcodes to ensure clear traceability, flexibility, and great confidence with no manual data entry. Divided into 3 separate acquisition groups with a calibration spot for each group. Each slide can be used for up to 3 times, once for each group, identifying 48 organisms per slide. Each box includes 32 MSDS target slides. So no cleaning required. It eliminates possible contamination that could lead to erroneous results or safety issues. Then each slide comes with 3 sections. So you have for a slide, you have 48 spots, but they're divided into 3 sections, 16 each, with 16 spots each. So it enables the flexibility of small or large sample run sizes. And you have up to 4 target slides can be run on the instrument at one time. So if you have 4 target slides, that is 4 by 48, that gives you 122 samples. So this is a very high throughput machine, okay? So let's say you have 16 spots, okay, that you want to disperse. You want to put your pure colonies and you want them to be dispersed so that you can get your mass mass spectrometry chart. Then you're able to get this result in 20 minutes for 16 spots. Okay, so this is a very high throughput techniques that with the shortest turnaround time that you can use concurrently with running up to 192 samples at one time. Okay, so just to mention something, because this is the last technology that I'm presenting on the. Let me go back. On the indirect identification methods. That is the last technology I'm sharing with you. So you are going to notice there is something consistent in both of them. You have a database, okay? So for API, for Vitek, you have a database, okay? Different companies, the research and development teams, they come with different databases. So in these databases, like in API, you have profile numbers that are specifically tagged to an organism. For Vitek, you have a bio number generated from the 64 biochemical tests that I've just shared when you're trying to identify your Enterobacterial C. So that bio number is now checked in the Vitek database for you to come up with a specific ID. Then you have mass spectrometry, which will measure the mass to charge ratio of different proteins in a microorganism to come up with a spectra, which can then be checked in a library of spectra or a database of spectra to come up with a specific ID of an organism. So they all clearly rely on the isolation, on the cultivation and isolation of an organism. And then that is what you introduce into these systems to give you give you a specific organism. Now, some are faster than others and even more accurate than others. So API, like we've said, you have different API profiles. I've just talked about 20E. You'll have API for Staphylococcus, for Streptococcus. You'll have for non-Enterobacterial C. So you'll have different profiles. Sorry. Let me just get some water. Sorry, give me a minute. Yes, so you'll have different indices for API that will give you a profile number that is then checked on a database. However, you'll have to maybe if you're trying to negate, let's say you have a lactose fermenter, and a lactose fermenter, you don't have an oxidase disk. So you are fully automated, okay? So API will have probably a limitation there because you'll have to have a profile an API index for an Enterobacterial C, and then you'll have an API index for non-Enterobacterial C. You understand? So with a Vitek card where you have 64 biochemical tests, then you'll only need one card with the 64 biochemical test to help you identify your bacteria. So it it cancels or it merges it merges the different API profile indices into one. Okay, so for maltitol MS, again, we've just talked about it. It will give you your organism, as long as you have your pure isolate, you can have your organism in 10 minutes. So the idea is to reduce to reduce the time when a patient has come to the hospital and they have gotten their sample into the laboratory and they have started empiric therapy. So you're trying to reduce the time from empiric therapy to a guided therapy. Okay, so the faster you can identify an organism and even give a report for this organism, the better a patient is managed. So these are just how these technologies help us reduce that time frame, and this is very important, especially for antimicrobial resistance. Okay. Then we have molecular techniques. Like I said, these are direct method. I listed microscopy, and I listed microscopy and molecular techniques under the direct methods, because in this case you do not need to cultivate this bacteria. You don't need to culture it or come up with an isolate. You just need the sample. So for molecular techniques in bacterial identification, they rely on analyzing bacterial DNA or RNA to determine their identity. These techniques offer high sensitivity and specificity, often bypassing the need for traditional culture-based methods. So I'll mention the key methods like PCR and real-time PCR and multiplex PCR. So for real-time PCR, I know many of you have interacted with GeneXpert. So the GeneXpert system is a real-time PCR platform developed by Cepheid for automated and rapid molecular testing. It integrates sample preparation, nucleic acid amplification, and detection of target sequences in a closed cartridge-based system. The system is particularly known for its use in diagnostic TB and detecting rifampicin resistance, but it also supports testing of other pathogens. So that basically GeneXpert uses real-time PCR as the molecular technique. And then we have syndromic testing that uses multiplex PCR. So multiplex PCR syndromic testing is a diagnostic approach that uses multiplex PCR to identify multiple pathogens simultaneously from a single sample, often in the context of a specific syndrome or set of symptoms. This method is faster and more comprehensive than traditional testing methods, enabling quicker and more targeted treatment decisions. So you'll have different profiles for different syndromes, and yeah, so I can mention maybe Biomiro that uses multiplex PCR as the principle for its molecular identification techniques. So then, now that we know all these techniques, what are some of the considerations that we make to select the different methods that we are going to use? So I differentiated this in terms of technical considerations and management considerations. So for technical considerations, this will be mostly, your benchmark, the type of organism suspected, the sample type and quality, the sensitivity and specificity of the method, the turnaround time, and how do you interact, is it feasible to integrate this method with your SOP and your LIS. So these are the technical considerations. Your management considerations will be the availability of equipment and resources. Is your region access? Can your region access different automated technologies? Do you have automation technologists? Do you have trained personnel? You also have to check cost and effectiveness. You have to check regulatory and accreditation requirements and also the purpose of identification. So is your setup a diagnostic setup? Is it a surveillance setup? Is it a research setup? Or are you investigating outbreaks? Yeah. So for the conclusion, the tools for determining the identity of a microbial sample have been emerging in the last decades. Although having limitations, culture and microscopy are still two of the most utilized techniques. PCR and other genetic approaches are particularly important for non-culturable organisms. So mass spectrometry has been shown to be a useful, quick, and easy for the identification of microbial samples and detection of microbial threats. However, it is reserved for pure isolate and cannot be used for complex samples since they may promote interference in the background. So in the future, development of detection limits of microorganisms will continue to be a key assignment in clinical microbiology. The combination of these methodologies and instrumentation will surely improve the skills for the detection of pathogens. So that is the end of my presentation. I hope we were together and that we have appreciated the transition of these identification methods from the conventional to the novel technologies. Yes, thank you.
Thank you, Madam Michelle. That was wonderful. We really appreciate the knowledge that you've impacted to the over 2,000 participants whom attended this webinar. And you also want to look at the question and answer section so that you can also answer a number of questions that will come as the members are asking. So we're looking at our first question, and the first question is, let me see.
Can I stop sharing my screen?
We're looking at the first question.
Sorry,
And
sorry, just a minute.
yes.
Can I stop sharing my screen so that I can see the questions? Is that okay?
That's okay. That's okay. That's okay.
Just a minute, please.
Okay.
I hope I have stopped now.
Yeah. So are you ready so that we can tackle the questions?
Yes.
So let's look at our first question from Lois Ayuma. And she's asking, what's the difference between manual and automated phenotypical identification in terms of time, cost, and quality?
Sorry, let me just write that down. I don't know why I can't see. Kindly, I can just see we can't see the slides not moving. I want
But
to
you are able to
access
see
it.
the Q&A section. Are you able
Yes,
to see the Q&A section? Are you
yes, I'm
able to
able
see
to
the
see.
questions that are in that section?
Yeah, sorry guys, the slides
Uh,
were not moving. Sorry, I'm just seeing slides
okay.
not moving. Okay, I've gotten Lois's Ayuma's question.
Yes. So yeah.
So what's the difference between manual and automated phenotypical identification in terms of time, cost, and quality? Or are we answering them one by one, or are we taking all of them?
Yes. One by one.
In terms of time, sorry, somebody has answered. Somebody has kindly. Somebody. Okay. When anytime somebody asks a question, the tab moves, right? So I'm not able to see her. In terms of times, please read it
In
again.
terms of time, cost,
Time,
and quality. Cost and quality.
cost, and quality.
Quality, yes.
Okay. So just to answer that, your manual identification methods in terms of quality, there are so many moving parts when you're doing manual identification. Okay. When you're doing QC, you have to QC all these moving parts. okay, when you're doing manual identification. So many laboratories may not have the resources to ensure you are actually QCing the manual processes. Let's say you're trying to identify E. coli, you're going to probably use a TSI method. You're going to use probably IMVIC and all these manual biochemical tests, and then you have to QC all of these biochemical tests, and all of these biochemical tests, they have specific QC requirements. Okay, so you'll have to QC them against non-microorganisms, like let's say ATCC strains or NCTC strains, to help you ascertain the quality of these processes. Okay, so this can be very costly. Okay, to ascertain the quality of your manual processes. So you'll find you will do them and maybe disregard the quality aspect of it, and therefore this has of course this will have a direct implication on the quality of your result. And then also in terms of quality, most of these manual identification methods, let's say you're doing a manual identification biochemical testing, they're usually very color change specific, a substrate specific color changes that could is different between a TNO and and Jerry, you see. So if maybe probably you're alone in the lab, you may have limitations in terms of making out what is positive and what is negative. They are very reliant on your competency and your background knowledge. Okay, so like I've said, so many moving parts. In terms of cost, of course, your manual identification methods will be less costly. They will be less costly. They're not as expensive. In terms of time, they are more time consuming. Let's say it's a test like citrate utilization test that could go up to 3 days. You going there and checking for color change, going back and checking for color change. That is 3 days. And citrate utilization is a biochemical test that is used to identify a very big family of gram negatives. So that means you'll be needing it for most of your biochemical processes. So that will in fact increase your turnaround time. So I hope I've just answered that. So if you're in the long run, if let's say if your business model as a laboratory is highly dependent on turnaround time, okay, then you'll have to weigh this, and obviously your manual identification methods may not be favorable for that kind of a clinical setup. So I hope I've answered Alice.
Yeah, thank you. Let's look at another question from Anonymous, and it goes, why are the agar plates put upside down for bacteria while upright for fungi?
So for bacteria, you want to put your agar plates upside down for bacteria because the condensation that could occur if you put it upright, you could have the top of your petri dish. It could have condensation, and that could create droplets and into your into your inoculated organism. Okay. So for fungi, you'll find most fungi, they grow, they're filamentous, okay? So they will grow upward. So then you'll have to put it upright. I hope I've answered that. I don't know if maybe you're muted. I hope I've answered that. So your fungi, you'll have filaments that will need to grow upwards, your spores. So you'll need to give that, you need to have that very upright, you need to incubate the petri dish in an upright position. For bacteria, you're afraid of maybe the condensation could get into your plate, creating contamination and so on and so forth.
Thank you. Another question from Steven, and it goes, what is the confidence range of API in identification of an organism?
So the confidence range of an API. So this we've said who, Stephen?
Yeah, question from Steven Haemba.
Okay, Stephen, the confidence ranges for these specific methods can be explored with the R&D research research results. Okay, so you'll find that if you need that report for the sensitivity and specificity in comparison with different methods, then you can get that from, I think they are openly available from the R&D, maybe R&D category of these specific technologies. So I cannot say off the top of my head what the findings were. Okay, and then you'll find that for different organisms, you will find different sensitivity. Like you said, API, the R&D team said for API 20E, the confidence level for E. coli is at 82%. For API 20NE, the confidence level for Pseudomonas is at 88%. And then, yeah, so this information I may not have at the top of my head, but yeah, they are available with the manufacturer of these different technologies.
Thank you. Kindly specify the measurement of standardized bacterial suspension.
So bacterial suspension, that is when you want to do your antimicrobial susceptibility testing. And also, okay, the standardized methods are different for different organism. So what we're using for aerobic organism is 0.5 McFarland standard. I hope I've answered that. For yeast, you'll have a
Thank you.
different standard and so on and so forth.
Yeah, thank you. Peter Kionde is asking, in API system, how do we handle the last two biochemical tests? That is NO2, N2, MSC off.
How do you handle the last two biochemical tests? Okay, let me just come back. I'll come back with the answer to that.
Okay. Another question from the same. In API, is it advisable to use any culture growth colonies?
Yes, in API, is it used to? Is it? I thought we were continuing the question.
In API, is it advisable to use any culture growth colonies?
Culture growth colonies.
Yes, that's the
I
question.
don't understand what he's saying. Okay, for API, like I've said, remember we have said these are indirect methods. So I've talked about API as a biochemical. It's an indirect method, meaning it's culture dependent. Okay, so you have to actually do culture, and then you come up with your isolates, and then you introduce your isolate into the API system. Okay, so you have to have your culture plate ready. Make sure you have pure colonies, pure single colonies, and then you make your 0.5 McFarland standard, and then you introduce it into the API cupules that are impregnated with substrates. So I hope I've answered that. you have to use pure colonies from your previously cultured plates.
Thank you. Mark Juma is asking, kindly elaborate more on Vitek MIC probability percentage.
Okay, who is asking? Sorry.
Juma, Mark Juma.
Mark Juma, I think, Arthur, if you give me another platform for this, I will handle the antimicrobial susceptibility testing part. So, like I've said, I wanted this PPT to help us smoothly transition from identification to antimicrobial susceptibility testing. So, what Mark Juma question is in minimum inhibitory concentration, which is a part of the antimicrobial susceptibility testing. So he says to, right now I could maybe answer it. Sorry, he says to answer what?
Kindly elaborate more on Vitek MIC probability percentage.
Probability percentage. I may not probability percentage. I don't know what he means by that, but I will have more information on MIC in a different PPT presentation. So I'll just write it down. I don't know what he means by probability percentage. Oh, okay. Sorry. So the confidence level for your probability percentage are in your ID. That is when you need your confidence levels in your ID. Your antimicrobial susceptibility testing when you're using MIC is for this particular organism, what is the lowest concentration that it's required to clear this particular antimicrobial agent. Okay, so your probability percentage will will cease at the ID level, and then from then you'll do your AST. So you usually need a 95% confidence level before you can proceed to your AST. Or if you're using it concurrently, ID and AST, then you need very, very pure colonies.
Thank you. Alvin Kinji is asking, at point, do we require CLSI or outcast charts?
Yes?
At point, do we require CLSI or outcast charts?
At what point do we require? Okay. So that is in the antimicrobial susceptibility testing. Like I've said, this was just an identification PPT. So we can explore your CLSI, your UCAS charts on a different platform for antimicrobial susceptibility testing. So that one you'll need after you've identified your bacteria and you're now trying to know the sensitivity of this, you're trying to check the sensitivity, resistance, intermediates, outcomes for different antimicrobial agents. So that then you'll require your CLSI to guide that.
Thank you. Another question from Benedict Wachala. Due to resistance of bacteria to antibiotics, what is the solution to this menace?
Yeah, like I've mentioned here, the solution will be it's different ways of looking at it. It could it will range from policy making, okay, where you'll have to check the antimicrobial agents that are allowed in the country. You know, there is the policy making process. There is your role as a clinical laboratory, your role as a medical laboratory officer working in an microbiology laboratory. Okay, so everyone has to play a role to counter antimicrobial resistance. Okay, so like for the purpose of this PPT, the role of the IVD in vitro diagnostics is to help us shorten the time between empiric broad spectrum therapies to more targeted therapies. Okay, so you'll find that different technologies the best technologies will try to reduce this time while ensuring the results are still reliable. Okay, you as medical laboratory officer, there's several things you can do. I can say, for one, you can give advisories on you can do client advisories, what drugs to take for what specific organisms, that are those of course are heavily dependent on your laboratory result outcomes. Okay, somebody's requesting for this specific drug, it's not tested for it is not tested for this particular organism. You have to advise that and even redirect that. The fast communication of your gram stain reports for blood culture, so everyone has a role to play, from the government to the pharmacist to the laboratory to the management of your different facilities. So everyone has a role to play.
Okay, Lawi Mwangombe is asking, is saying, Vitec is a game changer in identification of microbiological organism, but its costs and effects on TAT has led to underutilized. Please elaborate how to upscale use of Vitec in our county laboratory hospitals.
Sorry, you might need to please repeat that question.
Yeah. Let me repeat the question now. Vitec is a game changer in identification of microbiological organisms, but its cost and effects on TAT has led to underutilization. Please elaborate how to upscale use of Vitec in our county laboratory hospitals.
So from what I'm saying, according to who? This is
Lawi.
who?
This is Lawi.
According to Lawi, Vitec has a negative cost implication and a negative turnaround time implication in that specific facility. So is it the cost in terms of cost per test? I don't know. I don't understand what maybe Lawi is saying. Maybe because I know the cost you'll incur when you're doing when you're actually trying to get this machine into your laboratory. Now you'll have to chuck some change for sure. There are other maybe arrangements that you could do with the distributor or the manufacturer to mitigate that. When it comes to turnaround time, I could say the turnaround time is quite short compared to the manual methods, okay, for Vitec. Okay, you'll have the best turnaround time for Vitec. If there are some IDs that take 3 hours, okay, before you get your report. Some take 5 hours for identification. Then for AST, you can get your report within 18, sometimes even shorter number of hours. So I don't know how that Vitec has a negative implication to the TAT of that specific facility. So maybe they could just know why they're not utilizing that machine, and they could sit down as management to see what's wrong. The only thing that could make you maybe not give your report on time is if you're using mixed colonies and you're not, you're wobbling in the dark and you're trying this and it's not working. So you have to have pure colonies, and that has to stem from your culture, your gram stain, your purity plating, so that you have the shortest turnaround time. So he's talking about upscaling something.
Yes.
Upscaling what?
How to upscale the use of Vitec in our county laboratory hospitals.
Maybe you could reduce the cost if the cost to the patient is high. you could maybe sit down with the management and see if you can reduce the cost. But I wouldn't know. Maybe you could, yeah. The less closely it is, maybe, and then you could do advisories to your clinicians to utilize culture and sensitivities and not heavily rely on empiric methods. Okay, so for county and public health settings, you'll find that you'll have empiric guidelines, so you're not really keen on doing your culture sensitivity identification and susceptibility testing when you have empiric guidelines that says if this person is presenting this way, you put him on this. Okay, so maybe you could start doing advisories to your clinicians, your doctors, to have a more positive approach when it comes to utilizing culture and sensitivity in that facility. Hope that's okay.
Okay, good. Another question from Eric Langat, and he's asking, how about setlytism test?
What?
Setlytism test.
I don't know. Maybe let me find that in the comment and see what he's talking about. Set what?
Setlytism or setlytism test.
I'll write it down. I'm not.
Yeah, as we wind up, maybe you can tackle maybe the last question because of time. And you can tackle the question of Nicholas Kurgat, and he's asking, in terms of cost between MALD off and Vitek, which one is more economical in public facilities?
So, sorry. So, in terms of Malditov and Vitek, which one is more
economical in public facilities?
So,
yes.
Vitek will give you ID and AST. Okay. It's inbuilt to give you an identification that uses biochemical tests, and your AST that uses antimicrobial MIC, minimum inhibitory concentration, microdilution methods to give you your AST result. Malditov MS will give you ID only. Okay, so if you're trying to integrate the two, okay, those machines are both very expensive. So the Malditov MS is more expensive compared to the Vitek 2. However, on the cost implications, you're now running the equipment, you'll find that integrating identification from Malditov MS and AST from Vitek, that will be less costly compared to using Vitek exclusively. Okay, so if you're using Vitek exclusively for your ID and AST, that might cost you more compared to when you're using Malditov MS for your ID and then moving to Vitek for your AST. Okay, so now the cost implication will be in the initial phase of the installation. So yeah, I hope I've answered that. I don't know if I've veered off, but I hope I've answered that. In a public health facility, there are so many moving parts. I may not be the best person to advise in such in such setups, but yeah, the best you can do as an MLO is probably to just give your advisories and see if they can be accepted. You give your advisories to your superintendent or whoever is in charge, and you see if they can be accepted. So you can delve into the cost issues with the distributors, but what I can confidently say is integrating ID from Malditov MS with AST with Vitek 2 will be cheap compared to exclusively using Vitek 2 for both your ID and AST. Now the installation, Vitek MS will your Malditov mass spectrometry might be more expensive, so you have to weigh your options there.
Yeah, thank you, Madam
Sorry.
Michelle.
Let me just answer. Willie Sochanda has a good question here. Willie Sochanda says, good presentation. My question is, are you using any of these novel technologies such a lab? If so, is it possible to have video presentation of these techniques for better understanding? Yes, I don't want to sound like I'm marketing, but yes, we're using these novel technologies at Metropolis. We are using biofire field. We have biofire film array. We have Vitec 2. We have Maldito FMS. Yeah, we have all of these technologies because yeah, we are very big on turnaround time, and whatever technology is there that gives the best output at the shortest time, then we will get our hands on it. So thank you, Willie Sochanda, for that.
Okay, thank you, Madam Michelle. We will wish to allow you give us your parting shot. Because of time, we are past our normal webinar duration, and so maybe you can give us your parting shot as you adjourn this, as we adjourn this particular presentation, so that we can do another presentation in another day when scheduled. But for now, we kindly, you can give us your parting shot, and then I'll give the way forward. Thank you.
Okay, so yeah, thank you so much for being with me during this presentation. So, for if I've not answered you adequately and you need clarification on a few things, you can reach out with your questions through the host. But I've really appreciated your time for being here with me, allowing me to share my knowledge and understanding in bacterial identification methods. My wish is that you will have the same forum for antimicrobial susceptibility testing, where we can now see the novel technologies that are also in that aspect of clinical microbiology. We can also delve on the conventional methods as well. So I want to thank you so much for your time. And yeah, Arthur, thank you. Arthur and your team, Felix, and LMSK as a whole. Thank you for allowing me to use this platform. I don't know how many attendees were there, but I hope that whatever I had to say has gone to a very big audience and can be utilized. So thank you.
Thank you, Madam Michelle, for that. Actually, you presented to a participant number of 2,000, and so I believe a number of them have benefited from your presentation today. Some of them are requesting if you could maybe share with them the presentation in terms of soft copy, and so we will find it. We will maybe consult you, and if you allow us to share, then we'll be able to share this presentation in soft copy so that they can also utilize further, so that they can also learn much that they were not able to learn during this presentation. Otherwise, we are so much grateful and thankful for you coming to present with us at LMSK. To our participants, also thank you for choosing LMSK as your learning platform. And we hope and believe that on Wednesday you'll again join us as we do another webinar, same time 3 to 4:30 p.m. And for now, you'll allow me to end this session and meet you again on Wednesday, same time. My name is Arthur Imari, the LMSK Secretary General, and I wish you a good evening. Thank you.